Hello guys, my name is Dr. Varun and here we are back with 30 days, 30 most important topic for NEET 2025 biology.
Chalo, let's complete it.
Well, NEET is just around the corner and one of the very important topic from which highest number of questions and especially the statement based question can come to you. Scientific discoveries based questions can ask to you. That is from the molecular inheritance of genetics. Okay. So here we're gonna be starting with that chapter. The first thing in the chapter, okay, something we're gonna be writing it out. Sometime I've already written it so that I can squeeze this video as informative as possible. Okay, then DNA. We know that DNA is what? It is a polymer. Polymer means multiple units of deoxyribonucleotides has been used into it. If we are talking about RNA, then it is what? Polymer of ribonucleic acid, not deoxy, because at 2 dash or 2 carbon position OH group is present in the ribose but not present in the deoxyribose. Moving on, these 4 example match the following can come to you. You have to remember that this bacteriophage has what? This number of nucleotides. I'm saying that nucleotides, not the base pair, means it has what? Single stranded molecule. Then bacteriophage lambda has 48502 base pairs, means if you multiply by 2 you will get what? Nucleotides number. So this way the question can come. This whole thing, this is a haploid cell given to you, means it is talking about either sperm or ovum. Okay, they're not talking about the diploid cell. If diploid, so you have to multiply by 2. Okay, next thing is structure of polynucleotide chain. So over here if you see that this is how a long chain will look like, and if I had to mentioned it what it's like different bonds that are present. So we know that nucleotide has what? One nucleotide has 3 things present. What are the 3 things? Number 1, sugar. Which sugar arises like attached is what? Your phosphate, and there is attachment of what? Your nitrogenous base. So this A is representing what? It is representing your nitrogenous base, this phosphorus, and this is your sugar part, and this is making only 1 nucleotide. 1 2 3 4 nucleotides are keep on adding up. So we have to remember certain bonds and certain things which are present over here. So if you see that if you are able to see that this is what? This is a carbon of the sugar. It is making a bond with the phosphorus. This bond name is what? Phosphoester bond. Okay, so this bond name is what? Phosphoester bond. Then the next bond is present between this and this. Can you see that this is phosphoester bond? Similarly, it is making bond with this carbon also, which is the second number one. This is the first carbon, second carbon, third, fourth. Sorry, 1, 2, third. This is third, fourth, and fifth. So on the fifth also it is joining the bond, that is called as another phosphoester bond. Since 2 phosphoester bond is being present here also and here also, okay, we call it 2 phosphoester bond present. So 2, we use the word what? Di. So this bond is what? Phosphodiester bond. Okay, and if you see this another bond which is present between nitrogenous base and the first of the sugar, that is known as what? It is known as N-glycosidic bond. These bonds you have to remember. Next thing you will remember that its polarity. We said that the DNA is anti-parallel. This and this will be opposite, okay, when they are present together. How we able to find it? We actually find out that how with respect to the sugar only. Sugar is what? This is the sugar. Now we have to see that if the sugar over here, if it is it is making a bond with phosphorus, but here the phosphorus is free, means this sugar can further extend it into the next one. So means which sugar part? This is the 5th carbon of the sugar. So this becomes what? 5 dash, because it is still available to join with another nucleotide. Whereas if you go on the end of over here, if you see this one, this is what? Third number, 1 2 third. So this third number over here is free because it can make bond with another phosphorus. So we call this end as the 3 dash end. This becomes the polarity of the DNA molecules. Moving further, if you see that in this whole of this thing is constant. Only this thing is being changes, nucleotide base. Since it is a constant, we call it is a backbone of the whole thing. Okay, same. Nitrogenous bases keep on changing according to the different pairs that is coming up. Okay, then the next thing is basic things, that is, we have to remember that the Frederick Mischer identified the acidic nature of it and named it what? Nuclein. Okay, so you have to remember that nuclein. Okay, then in 1953, what happened? James Watson and Franz Crick, they give what? They give the double helical structure of beta DNA. Double helical structure of beta DNA. And on which person's research work they have given up? They have given up on the work of Maury Williams and Rosalind Fleckens, okay, and Franklin's, and they did experiment of what? X-ray deflection, है ना? Yeah, X-ray deflection technique. Through that they find out the composition, okay, and later on there is a person called as Erwin Chargaff who gave what? He gave the Chargaff rules. In Chargaff's rules, what happened? In Chargaff's rules, he told that the amount of the adenine and purine, how much it would be. Sorry, purine and pyrimidine, how much it would be. So he gave that purine will be equal to pyrimidine, and if we will be writing like then it will be like purine upon pyrimidine will always be 1. But the bonds, like A will make bond with what? T and double bond, and C will make triple bond with G and vice versa, and their number won't be equal. Means this, if A plus T and G plus G, if you taking, it won't be equals to 1, and it is variable for different species but constant for a species. Constant for a species, it won't change. Let's just see that it's DNA molecule briefly. Then in the DNA molecule, this 2 strands I have made. Okay, we can give it a polarity. So imagine that this is 5 dash. So other end would be what? 3 dash. Here it will be 3 dash. Other end would be what? 5 dash. Then they are making bond. A will make a bond with T with the double bonds. G will make with the 3 bonds with the C. That is known as what? These are the hydrogen bonds. So one another bonds that we have to remember, that is the hydrogen bonds that are occurring here. Moving on, if you see that between these two, okay, over here, between 2 base pairs, there is a space present. How much is this space? You have to remember 0.34 nanometer. In nanometer given in NCERT, if you change into Armstrong, you have to do the -10. Okay, so next moving on is the what, like if you see over here, from this to this, it considered as 1 pitch. Okay, so this 1 pitch, it contains how much? 10 base pairs. Pair means 10 means 20 nucleotides. They are in the pair form, and this much is how much distance? It is 3.4 nanometer. 1 pitch. These things you have to remember. Question can be asked to you. If they ask like between the 2 strands, they are moving like this. What is the difference between these 2 strands? Then difference between these 2 strands is 20 angstrom. This is the basic features which has been told to you. Moving on to the next thing, that is central dogma. Central dogma says that when a cell is working, how it is working. DNA has all the information, but DNA is not directly working. It is someone other, it's a protocol kind of a thing, that highest level, then this. It's simply like what? You have a principal give it to the headmaster will give it to the teachers and make teachers do the work. That happens same over here. So here principal is what? Your DNA. DNA will give it to the headmaster. Headmaster is what? Your RNA. So DNA will make RNA. That process is called as what? Transcription. Moving on, from headmaster it will go to the teachers, means RNA will make the teachers. Teachers are what? Proteins, which will do the work. So this is called as what? Translation. Now, if the cell is dividing, cell has to make again another DNA. This process is called as what? Replication. Sometimes in certain viruses whose genetic material is RNA, so when they go and infect a DNA containing organism, then they change their RNA into DNA so that their DNA, like the viral DNA, can go and attach with the DNA of the host. So they have to do this process. So what they will do, they make their RNA into DNA as it goes inside it with the help of enzyme called as reverse transcriptase, and the process is called as reverse central dogma. So this process is what, like whole system is called a reverse central dogma, and this process is called reverse transcription. Okay, these two things. Then the next thing is the packaging of DNA. Now DNA is what, imagine that how long a DNA would be. If we started calculating, then it comes out to be more than 2 meter long in the one cell. But one cell is microscopic, how it is taking a 2 meter long thread. Then the 2 meter long thread is packed, so it is coiled, means just imagine like that. You have seen that with the tailor, they have what, a thread stick, and thread is around it. If you open it, the thread will become so large. So the same thing is opening, like taking place over here. Okay, so here they are packed inside it. So how they are packed inside it? See this is the DNA molecule. First it is packed around what? It is packed around these circular bodies. These are what? These are the histone proteins. So these are what? These are histone proteins. And they do what? They are actually 5 different types of histone proteins. 4 works into it in the pair of 2. Okay, so means total 8 of this comes together and forms what? Octamer. This octamer is also called as new body. Now around this new body, can you see that this green color thing is what? This green color thing is the wrapping of the nucleotide, R thread. So the DNA is wrapped around it. Around 200 base pairs are wrapped around it. Almost like 1.75 or 1.65 turns it takes. Okay, or 3/4 turns also written in somewhere. Okay, so remember it. This whole structure is called as what? Nucleosome. Okay, so what do we call it? We call them nucleosomes, which has already been mentioned. Now nucleosomes are made up of histone proteins. Okay, this we have studied. And these histone proteins are basic in nature. Means if the proteins are there, means they must be made up of amino acids. Means basic amino acids are needed to make a basic protein. Simple fact. So what are they? Arginine and lysine. Okay, and you have to remember this fact from the biomolecules chapter also itself. Okay, coming back, the next thing is this nucleosomes come and form a pair of 6 like this and further coiled. This is called as what? When they are present in the pair of 6 comes together and coils and keep on coiling, 6 nucleosomes. It is called as solenoid. From solenoid, it forms super solenoid. This much. Next, during the time of division, cell is dividing, it further condensed with the help of non-histone chromosomal proteins. During the time of division only, during cell division. Non-histone chromosomal proteins, because cell size will decrease now when it will take place. Okay, so we have to further condense it. So what will happen? This non-histone proteins will do what? It will condense it into chromosomes. That much you have to remember. After this, the next thing is the histone proteins and non-histone proteins. We have already discussed about it. Then non-histone protein, and then the next thing is that how the DNA molecules would be present. Like it will be present in the 2 forms. That would be what? Either it could be transcriptionally active or inactive. Transcriptionally active means it will make RNA. Transcriptionally inactive means do not make RNA. When it is transcriptionally active, it will be lighter on dye. Okay, and if it is darker on dye, it means it is transcriptionally inactive. It is transcriptionally active, means it will make the RNA. So the lighter one is called as euchromatin and it is called as heterochromatin. So we're gonna be more talking about the euchromatin now in the next subsequently part. But before to start it, how do we know that DNA act like a genetic materials? So we have a three different experiments. Okay, number one experiment is the transforming principle by the Griffiths, given in 1928. In this experiment, he took what? He took the two different strains of the same species of a bacteria. Okay, in that what it does, one is the virulent, other is non-virulent. When they give the rough strain, which is non-virulent, it injected into the mice survive. Means this bacteria do not cause diseases as expected. When it gives the smooth strain, which is virulent, means causes diseases, give it into the mice die. So we know that this has something which is making it what? Making it bad. Okay, now he did what? He heat killed this smooth strain, and when he heat killed it and give it, then the mice survive. Next what he did, he gave the live this and this. Normally in both the cases, if it gave this, it should alive. If it give this, it should alive. But when it gives this, then what happened? Mice die. And we inject out the things from the mice. We found that live smooth strain was present. So Griffiths said that the R strain take up some of the material of the dead thing and transform itself. Okay, so this is what he didn't told what it is, but it was believed it is proteins it has taken up. Okay, next thing is what? Then the second experiment, which was the further enhancing the understanding of transforming principle, they did what? Biochemical assay of it. What they did, they take the material which is suspected to be or can be the genetic material. So proteins, DNA or RNA. So in one case, what they did, they took the heat killed S strain. Okay, and in that heat killed S strain, first they put protease enzyme. Protease enzyme will dilute all the proteins present in the heat killed, and they give it to the R strain. R strain transform, means S strain appears. Means what? Okay, we destroyed all of the protein, but still they are being changed into a something which was what? Transforming. Means it's not protein, because protein we have already destroyed it. So it is something else, either RNA or DNA. In the next, they took RNAs. RNAs will destroy all the RNA present in this heat killed S. Then they give it to the R strain. R strain again transform themselves. So means who is left? DNA. Let's see that if we kill the DNA, we put the DNase enzyme which kill all or destroy all the DNA. And when we give it to the R strain, no S strain appear. Means it was DNA only which was transforming. But not everyone will convinced with it. So means scientists are a little bit crazy. Okay, so they didn't accepted these three scientists' work. So you have to remember this. Then after around 1952, Hershey and Chase experiment came. In this, what they did, they used the radioactive technique. They used what? Bacteriovirus. Bacterioviruses are what? Bacterioviruses causes diseases in the bacteria. How they will create diseases? Of course, with the genetic materials to be thrown into the bacteria, then only they can do the diseases. Now they have only two things: either they have protein or they have DNA material. In one of the virus, they did what? They make the protein radioactive. In other, they make the DNA radioactive. They found out that the bacteria which has DNA will be present inside. Means something which is transforming, causing the disease, should be inside the bacteria. Bacteriophage is infecting the bacteria, right? So they found out that when they radioactive the DNA, it is present inside. So means what happened? Radioactivity is detected inside the cells. Means inside the bacteria. Means DNA is the material which is transformed or goes into the bacteria. Okay, the genetic material. So it proved like that way. Now these are the properties of genetic materials and how they explained it, given in NCERT. You can do it, remember it, question can come from here. So what are the properties? It able to generate its replica. Okay, it should be chemically stable and structurally also stable. Provide scope for slow mutation. It should not be no mutation occurring place. Environment is changing, mutation is not taking place, it's not good, it should change slowly. Should express in the form of Mendelian characters. So now there was a problem that these characters were more suitable for RNA. That's why RNA formed. Earlier in the world, RNA only formed first, but then DNA forms because DNA is chemically more stable, have slow rate of mutation. You know that RNA is not stable. That's why the viruses keep on changing themselves and we are not able to make the RNA viruses, vaccines, or medicine because they have changed themselves till the time we have made the medicine. So this was the thing. Moving on to the next thing, that is replication. Replication is what? Replication means you are making one another copy of the DNA. Now, when you are making one another copy of DNA, you have to remember that how they are doing it. By the Watson and Crick model only, we come to know that each strand will act like what? Template for the new strand to form. Okay, so this thing has been experimentally proved with the help of semi-conservative nature, that the DNA have a semi-conservative nature. Melson and Stahl did an experiment. In this experiment, what they did, first they grow the E. coli in a heavy nitrogen condition. So means they made their DNA which has a heavy N15 type of a nitrogen compound. Okay, so it's not a radioactive, it's a heavy compound. Heavy, like N14 is the normally present, N15 is the heavier atomic mass of it. Okay, so now they put it into it and they checked into the like what they are doing. So initially what they found the heavy medium only. So all the DNA would be heavy medium. So if you centrifuge it, you will see that it is coming at the very lower level because of the heaviness. Okay, now the next thing is they put what? Now they put it, the bacteria, into the N14. When they put it the N14, what will happen? There will be what? This DNA and this DNA will change into what? Another strand. But the next strand would be forming what? It will form N14. So it is a mixed DNA, means hybrid DNA. But this will be lighter than this one because this was 30. This is 29 only. So this will be what? Lighter. So you will see that something which is lighter present over here is here. Then they did again replication of it, and this time they found that N15, N14 separate. Now the two strands will have what? N15, N14, and two strand will N14, N14. So see this over here, what happened? This has what? Your lighter DNA. This has your hybrid DNA. So two different strands will come up. This is how they find it. Now on this basis, numerical can also come. Anything could have come. If you don't understand this thing, let me know that I will give a video for that also. Okay, moving on to the next thing is how the replication is occurring place. So over here, we will quickly see the different enzymes present and different things that is happening over here. Starting with what your DNA is a double helical. So what is it? That DNA is in the helical form like this. So to start the process, we have to open it. Okay, so that is what first step would be: unwinding of DNA helix. Who will do this? We have to remember the enzyme. So enzyme would be what? Enzyme is helicase. Next, second thing, after unwinding it. Okay, so next I am making over here that we have did what? We have opened it up. Now 3 dash, 5 dash, 5 dash, 3 dash. Next thing would be the recognition of the site from where we have to start. So recognition of the site of replication. Site of replication is always present on the 3 dash end. Now 3 dash over here, if you see, is present where? Here. And over here it is still binded. So closeness proximity is here. So 2' will come and join it. Now who will find this? This will be found by DNA dependent DNA polymerase. Okay. After this, once the site has been formed, what will happen? Nucleotides will keep on adding. Okay. So there will be what? There will be elongation. Of course, the primers are added. Primer added. Okay, elongation until what? Until this, until this. Now, as it reaches to this point, okay, as it reaches to this point, what will happen? Since it has to further open up, so what will happen? It will further opens up. As it opens up, what will happen? If you see that this has been opened up. So as it is opened up, what happened? This over here, the strand has been made. Over here, strand has been made in this direction. So this thing, the primer will say, 3 dashes there. So it will again go back at this site. Whereas this will keep on forming this thing in the first one. So this thing will happen. When the nucleotides are being added up, like this area, if you see over here, here what happened when you are opening it up, it created certain tension over here. So this tension has to be removed. So removal of tension will take place. Removal of tension. Who will do it? Topoisomerase. It is also called as gyrase. Okay, this one thing will occur. Now, removal of tension is done. The next thing is what? That when this thing has been opened up, there are chances that it can recoil, means the process is not completed. So we don't want to that. As well as we want this to be recoiling with each other. Okay, so these two has to be individually has to be recoiled with the new complementary part, the yellow color one that we have made. So this thing will be done by what? Another enzyme called as what? SSB protein, single binding proteins or stabilizing proteins. Single stabilizing binding proteins. Okay, so to stabilize the unwinded helix plus new strand helical formation. This will be done by SSB proteins. Next and the last one is what? That we will be going with what? We will be going with this strand. If you see this strand, what will happen? That one is forming this. So means these fragments are being formed. What these fragments are called? These are called as Okazaki fragments. Now we have to join these fragments. So joining of these fragments will be taken care by what? Will be taken care by DNA ligase. So this is what different enzymes. So remember what is helicase doing, DNA dependent DNA polymerase doing, topoisomerase, SSB protein and DNA helicase. DNA polymerase is also adding the nucleotides units also. Nucleotide units, when they are adding up, basically they are in the triphosphatic form and they changed into the monophosphatic form. When they are changing from triphosphatic form to monophosphatic form, they are what? They are releasing the energy by breaking the bond of phosphate. As well as the monophosphatic form, once they are forming, they are acting like a nucleotide substitute also for this substrate also. So means what? They are doing what? It is acting like the triphosphatic form is acting as a dual purpose in this process. This can be asked to you as an assertion reason. This is all about the DNA replication. Once the DNA is replicated on DNA, RNA will make. There are different types of RNAs present to it, where we won't go in deep because this is the easiest topic. Just go through is your NCRT, what are they, but important topic. So what are the different types of RNA? We have rRNA, whose formation is super important. Which is forming by what RNA? We will learn by that which RNA polymerase will form what type of RNA. Okay, that is rRNA. We have tRNA and mRNA. These three are the most important one because these three together collaboratively be helping in the process of translation later on. So this is most important. Now in transcription, the first question before going into the process and the detail, let's just see the first thing, that if they have given you a strand, okay, means if they are saying that this is the template strand, template DNA strand, make the RNA on it. Like what would be the coding transcripted on the RNA? So it will be complementary only. Okay, just only at the place of like instead of A making bond with the T, it will be with the U over here. Change means, for example, if the template stranded A, T, C, G, okay, like this three only. So its RNA will have what? It will have a complementary pair with respect to template. If the template strand they are saying, if they are saying coding strand, that it will change. We will learn that thing. So complementary thing will occur, means A would be what? It will be changed with its counterpart. A will make all in the DNA, it will always make a bond with the T. But here T is not present, it is replaced by U. So here it will be U. When it is T coming, its counterpart part A. A is also present in the RNA, so A will come. Then C, its counterpart is G. G counterpart is C. So this would be the code on the RNA. But if the question is that, what about the coding strand? If the coding DNA strand is given to you and then you have to make RNA, then the coding means what? Something which is being coded. Okay. Except at the place of T, it will be U. Rest all the thing which is present in code like DNA, it will be similar. So if, for example, A, T, G, C is present on the coding strand. So the RNA would be what? Same. Except at T will be with the U replaced. So A will be A, T will be U, C, G, like this. Very importantly, remember it. If the transcription is coming to you, then you have to see that whether the template strand given or the coding strand given. Okay, now the template strand, if this template strand will have a polarity, okay, so the RNA that will be forming, that will be 5' to 3' forming RNA, and this also 5' to 3'. Here the template strand will be this, here coding strand will be this. Remember it, here only fact, okay, that this. Moving on to the major process. Now the transcription has a 3 units present. What are those 3 units present? I am writing over here. So number 1, it has what? It has a promoter site. It has a structural gene. And it has a terminator site. Now if someone will ask like where promoter, where structural gene and where terminator present. So according to NCERT, we're gonna be learning the fact that is what your promoter, that promoter will be present on the, like it is with respect to coding strand. So this, if for example, this is the strand, this is your template strand, this is the coding strand. So template strand will be 3' over here, 5' here, 5' on 3'. So means where this structure is present, it is present here. This process is occurring here, means it is present upstream. So you have to remember that it is upstream at 5' end. Okay, 5' end of what? Of the coding strand. Okay, and this is present at the downstream at 3' end of what? Of the coding strand. In between there are what? Structural genes. That's it. Okay, this thing, if you have understood, let's go with the process. In the process, before to start it, we have to remember it that transcription in prokaryotes and eukaryotes has a little difference. Prokaryotes and eukaryotes. What different would be present? In the prokaryotes, their mRNA that is forming, it is polycistronic. P for prokaryote, P for polycistronic. But eukaryote, it is monocistronic. What does that mean? Polycistronic means from one mRNA chain, multiple different types of proteins can form. But the same thing is not present in the eukaryotes. So from here what? One RNA forms multiple proteins. But here one RNA forms one protein only. Now these cistrons are what? These cistrons are the sequences present on it. Okay, so there could be two different types of sequences present. One is what? One is introns, other is exons. So two types of sequences, cistrons are there. What are they? One is exon, another one is the introns. Now exons we need it because they only transcribe or translate. Transcribe is wrong. Translate. Where they do not translate, they are of no use. So we don't want it. Till here understood this thing. This thing will come more into the eukaryotes only. Prokaryotes nothing much. Let's go into the process. Okay. So when we are going with the process, okay. So this I am removing. Okay. Let's start with the process. So first of all, what will happen? Your promoter will come and join. Okay. So if the promoter has to come, so it has to what? recognize the site. So recognition of site. So this site will be recognized with the help of an enzyme. The name of the enzyme is DNA dependent RNA polymerase. RNA polymerase. It has dual function also, that it will help in the polymerase process also, in recognizing also. Now who will come and recognize it? There will be a protein, there will be a primer, there will be a substance. Okay, that thing has what? It has a certain structure present. So we call it what? We call it, it's a type of a holoenzyme. Now this holoenzyme has two parts. One is the core enzyme, which is made up of 4 different units, and on this arises a small extra thing that is called as what? It is called as the sigma factor. So this thing will, the sigma factor is important for finding the thing. Okay, this thing is important for the polymerization. Together they are important to go at the site. Okay, after recognizing the sites, the next thing would be the process initiation. Now to initiate the process, there would be what? There would be certain sites that would be identified. These sites in prokaryotes and eukaryotes are different. Okay, so that Tata box, Brigno box, these terms has been told to you, so that you have to remember. That will be identified. The process start and elongation occurs. Who will do the elongation? Same factor, like it will act like a dual function, all the substrates and all those things will be there. It will keep on doing it. As it reaches to the termination site, at termination site, what happened? There is another factor that come and join. That is a Rho factor. It will join and the process will be terminated. Join with what? Join with this thing. Meanwhile, when this process is taking place, after identifying, this sigma factor is removed. Because if the sigma factor still stay, that it won't lead to the smooth flowing of it. So that thing is given in your NCERT also, if you have seen this diagram. And this diagram here, you have seen the sigma factor, then the sigma factor is released and then the Rho factor has been coming over here. Means by the RNA has been formed. Now this process, the thing that has been formed, it is known as what? hnRNA. It is known as the RNA that is formed by this process is called as hnRNA. That is what? Heterogeneous RNA. Now the heterogeneous RNA is okay for the prokaryotes, but in eukaryotes further processing is needed. This is known as post-transcription process. In the post-transcription process, what will happen? We will process it into a mature form rRNA. Okay, so this mature form rRNA is basically what? Messenger RNA. Okay, so now this thing is what? This is known as post-transcriptional changes. So what we do? We do first is what? We add the head. On the head we add the cap, and on the tail we add a long polynucleotide chain. Okay, so what? Capping, not heading, wrong word, sorry. So what will happen? There will be capping and tailing will occur. In capping and tailing, what will happen? Basically at the head, there will be 7-methylguanosine. It is it's a nucleotide that will go and attach. Whereas at the tail side, what will happen? Polynucleotide, means more than 200 bases of the adenylyne chain will be added. This will increase its stability because RNA is very less stable. Okay, it will destroy soon. Second thing is that this in the eukaryotes, what happened? This is only taking place, both the things are in the eukaryotic, this post-transcriptional changes. There is what? There is the presence of exon and introns. So we have to do what? Splicing and editing. What is splicing and editing? It is like adding, if this is the part of the DNA which we don't want, we splice it away. splice it away. And then the left out part, we just join it and form what? mRNA. So this is the most stable form of the RNA, okay, that has been formed. This thing is there. Now with the transcription, what will happen? Genetic codons comes. Means what you have wrote the sequence over here about that. Now genetic codons and its functioning, work and everything has been the work of 4 different scientists coming together. Not coming together simultaneously, but their simple work has helped in is understanding this topic. So first thing is genetic codon is what? It is a sequence present on the mRNA which will be translated to make the proteins. Now George Gamiot tell us what? He told us that the codon is in the form of triplets. So he tell that codon is in the form of triplet. Okay. Then Har Gobind Khurana what? He proved it. Okay. How he will proved it? He proved that if the codon is in the form of triplet. So he did what? He make a homopolymer of protein and he make a heteropolymer of a protein. Okay. Now in the homopolymer, for example, he take U. This is a chain where only UU is present. And he take another chain where is UA. Heteropolymer means 2 nucleotides. Okay, dinucleotide only. UA. Now, if it is triple, let's take the example. If it is a triploid, then it is making what? This is one codon, another codon, another one. All the codons will be UU. Means all will be making what? All will be making the same amino acid chain. So when it was analyzed this whole chain was made up of what? Phenylalanine. No other type of amino acid came into the chemical verification. Next come the heteropolymer. In heteropolymer, what is happening? The first is this, then again this, then again this. Means there will be repetition of different. So this will make first type of amino acid. This will make different amino acid. First amino acid, different amino acid. But this and this will be same. This and this will be same. Okay. So it tells us that there are 2 different types of this amino acids. So when we analyzed it, we found it correct. So we said that it is in the form of triploid. So he proved it. Now when he is doing all of those things, for this process we need certain apparatus. Normally this RNA making a protein inside the cell. So someone must have helped us in finding it. Who was that? This person. Because this person did what? He made a process. Which was what process? The process was that cell-free protein synthesis. So he did what? Cell-free protein synthesis mechanism he has told. Synthesis. Okay. What for this to come on, there might be enzymes needed. So this person gave the Sivra Oka. He gave an enzyme. The enzyme name is phospholipid. Enzyme is the polynucleotide phosphorylase. Okay, that is what? Polynucleotide phosphorylase. This enzyme he gave it, which help in this process to occur. So they are both directly they didn't work, but they helped this Har Gobind Khurana, that's the Indian person, to finalize, proved this thing by the George Gamiot. They can be asked to you as the match the following or direct question. Moving on, then he said that genetic codons. Now you have to remember at least these genetic codons, which amino acid they correspond to. Now AUG is for the methionine. These 3 is for the stop codon. Then triple U's give the phenylalanine. Triple C will correspond to the proline. Triple A will be corresponding to the lysine. And triple G correspond to the glycine. This can be asked to you because lysine is in the basic amino acid which is a part of histone protein. It can be asked to you. Moving on to the SLAM features, they can ask to you. What are the SLAM features? Codon is triplet. 61 codes for amino acids. 3 codes for nothing. How many proteins which are needed in our body for the protein synthesis? That is only 20. Of course, more than that amino acids are present, but 20 are needed for the protein synthesis, whatever the protein in our body is needed. Then it is degenerate. Degenerate means some amino acids are coded by more than one codon. That's only the 61 codons are coding only for 20, means someone will be coding similar. Okay, so many doctors will be there like that. Unambiguous or non-ambiguous. Non-ambiguous means they are unambitious. Means one codon will code for only one amino acid. Means one codon, if it becomes doctor, means doctor only. It don't want to become anything else. Okay, for example, I am a dentist. So I am a dentist. I became dentist, after that I don't. But I am very ambitious person. I am not unambitious. Okay, so this thing over here comes. Then it is reached in the continuous fraction without any punctuation. Means A, U, G, A, A, C, A, A, G, like that way, without stopping anything. Okay, then universal. It is same for everyone except mitochondria and certain protozoans. Okay, then AUG is the initiator codon, codes for methionine. This is the stop codon, stops. UAA, UGA or UAG. This you have to remember. With this, codon is over. After that, the next process is translation. Translation is what? Protein formation. Okay, over here, when the protein has to be formed, we have to remember certain thing. So imagine, try to imagine. Okay, so what happened? We have your this mRNA. Okay, so this is our processed mRNA. On the mRNA, there is what? Codons present. So this is your AUG initiation, UUU, A, like this. Codons are being provided to you. CUA, like this. Okay. Now what happened? First of all, initially, just to see that what is happening. Initially, what will happen? That there will, of course, these will be identified and all those things are there. Initiation will be by the AUG. Now what will happen? That some, like, first of all, small unit of the ribosome, because ribosome containing what? rRNA. So I told you, rRNA will work. So ribosomes have rRNA. So small unit of rRNA will come and join over here. Okay, so small unit will come and join like this. It will join something like this, that two of the genetic codons will be coming together. Now what will happen? On this, there will be what? There will be, like, first one over here, I mean like AUG, there will be what? Corresponding anticodon present tRNA will come and join. So tRNA will join. Now the tRNA will not directly come like this. Before that, tRNA will do what? It will bring something with it. Okay, so there will be what? Initiation process. That initiation process of this will be what? It will be what? Prior to the initiation of translation, what will happen? Charging of tRNA with amino acid occur. And it occurs in two forms. First, what will happen? Amino acid will take the ATP and forms what? Activated. That amino acid is activated. Now this activated one will go and load on the tRNA. Okay, so here what will happen? For example, in the first case, AUG is there. AUG always correspond to the methionine. So what will happen? Imagine that this is the methionine. This is your methionine amino acid. So it will go and attach over here. Okay, so this is your methionine, first amino acid. Okay, but first I have to just make it space for it. Okay, now with this, what will occur? Now the next thing would be the larger unit of tRNA will join with it. Larger unit of rRNA will join with it. That is ribosomes that will join with it. Now over here, when it is joined, there is a certain spaces present. So this is a P site, this is the A site, and there is what? There is a E side also. Okay, fine, it joins. Simultaneously, what will happen? Or the second, like this position is free, A site is free. So on the A site, there will be what? Then another tRNA, which is charged. charged tRNA will come and join at A site. Now, if you see over here, then this UUG, that is correspond to what? Phenylalanine. Always remember it. So phenylalanine amino acid will be present over here. So another tRNA we will make. It will come and join over here. Okay. On this, what we have? We have phenylalanine. Now, as these two of them are present closer to each other, there will be what? There will be formation of peptide bond. So there will be what? Formation of peptide bond. And once this peptide bond is formed, what will happen? This bond will be broken up. So it will break up. So the tRNA that came with the methionine broke its bond and give it to what? Give it to the next one. And this whole structure which is present over here, it will move. So if it will move, how it will look? Let's just see it. So over here we have this whole structure. Okay, here we have our sites. This is AUG, UUU, AAA, CUA, and this is mRNA. So little bit small. Then we have what? We have this whole thing will move towards the next one like this. Along with that, this bigger one also move. This bigger one also moved. Okay, and when it moved, what happened? This spaces changed. Means what? Now P is here, A site is here. So means your tRNA which was present at UUU, means if you see that UUU, what is present? This is present. This tRNA with one this and with one first amino acid and the second amino acid joined. Whereas what happened with the first tRNA? The first tRNA meanwhile will exit. So what happened? It leaves from the E site. Exit. So E is the exit site. Meanwhile, the A site is again free. So another means third amino acid, another tRNA with amino acid will come and it will join here. Like this way, what will happen? This process will keep on going up and a long chain will keep on forming until there will be what? Stop codon. I hope little bit you would have understood from it. This is about the translation. In your NCRT, mutation is also given it, but I'm not taking it more because it's very easier one. Frame shift mutation, deletion, point mutation. So this thing you can do it on your own. So I'm not doing it. While this RNA that is present, there's a lot of areas present which are UTRs. That is what? Untranslated regions present. Means this thing is being translated, but still something present which is not being translated. It is to make this process more efficient that this thing only will translate, not this and this. Okay, so this thing you have to remember. For termination, we need a release factor that will be provided, that it will come and it will release this whole thing, this chain into a polypeptide which will do the effects. This is about the translation. After translation, next thing is that during which state we can actually regulate it. Means if we want to change something, we can do it. So the regulation in the eukaryotes can be done at the 4 different steps. What will be the steps? Number 1 is at the transcription level. When the transcription is being done from DNA to RNA, it can be done. Second, after transcription, this RNA comes out into the cytoplasm from the nucleus. This process is what? Translocation process. It can done. Even at the post-transcription process when it is occurring, Then time also we can change it. So at the post transcriptional level we can change it. And the last one is that at the like when the translation level, when the protein will make the when the RNA will make the proteins. So that is what at the translational level. But in eukaryotes we can't do it. In prokaryotes we can't do it. In prokaryotes, this step and this step do not take place. So there we have only first and fourth number step where we can change it in prokaryotes. Have to remember it. So the next topic is lac operon. First of all, remember that it is seen in case of prokaryotes. So it is a prokaryotic feature. Next point, that in prokaryotes what happened, not just single gene but multiple genes come together and do a single function. One such function is what? Lactose digestion. Okay, so in E. coli what happened, this lac operon is present. Now let's see that what is normally being happening when no lactose is there. So the first condition is what? No lactose in environment. So means E. coli is what? Simply without any food. Just imagine like that. So what its whole DNA doing? Okay, so its DNA, for example, the DNA would be what? It will have a double-stranded structure. This is your DNA I am making, that lac operon DNA. Now there would be a certain genes that would be what? That is collectively working. So I am making it over here, that collectively that those are working. So what are those? We have, this is the genes I am making, nothing much. Okay, so the first condition is these are the genes which are present on it, and how they are working we're gonna see. So when no lactose is present in the environment, at that time this I gene, it will make what? It make a repressor RNA. Okay, because this is what? This is a part of DNA. So it is making what? It is making a repressor mRNA. Now the mRNA will translate into what? Into the DNA. Into the proteins. So it will form what? A repressor protein. Now if the protein is formed, means it will do certain work. What work it will do? This protein will go and bind with this. And as it bind with it, what? It will stop to do any work. So means it will stop, because this is what? This is the operator. So operator is switched off. When this is happening, when no lactose is present. Now let's take it to the next step, when the lactose is present. So when the lactose is present, at that time what will happen? So all the thing I am showing you in the green color. Okay, so when the lactose is present, this lactose will do what? Normally this I gene will keep on making this. It will keep on making this. But here the lactose will work. Lactose will bind with it. Okay, so the lactose binds with it. Now imagine that if the lactose has bind the protein, will the protein be able to go and join with the operator? So it won't be able to join with the operator. So what will happen? Don't join with operator. And if it is not joined with the operator, then what will happen? This will be switched on. Switched on. And when it is switched on, what will happen? This ZYA will release what? They will make its RNA. And what RNA proteins it will make? It will basically the RNA will make what? It will make a protein or the enzymes which will help in digestion. So this you have to remember. The Z will make what? It will make beta galactosidase enzyme which will digest the lactose. The Y will make what? Permease, which will make the cell more permeable, that more lactose come inside. And this will be transacetylase whose function is not given. So that's okay. So now what will happen? As they will be released, what will happen? Lactose will be digested. Lactose will digest. That's it. So when nothing is happening, no barrier is there. So means these genes are doing what? These group of genes working as a negative. So that's why lac operon is considered as a repressor type of operon or the negative operon. That's it. That is lac operon. After this, the next thing comes is the Human Genome Project. From here, higher chances of questions coming. So I have already compiled up over here. So Human Genome Project is launched in 1990 and it completed 13 years. Till now also they are coding something, but most of the work is being completed. Okay, then approximately it is 9 billion US dollar program funded by these many organizations and the country. It requires like lot of information was produced. If this you will see, if each like there is a page, okay, on each page thousands nucleotides can be added, then we need a book with 1,000 pages. And such books, how many books we would need? We would need 3,300 such books to compile it. So we need certain field which can store this information, and we made what? Informatics. Okay, now the next thing is that is just the human beings who were taken? No, these all the other thing also which were also we did the later on genomic projects, means we find out that what is how they are being there. So this you have to remember. Over here a nematode name is being given to you. Nematode means they are belonging to non-helminthes or aschelminthes. So except those 3 examples which are given in NCERT Animal Kingdom, 2 are given in 12th class, one here, one in the RNI information, okay, biotech. Then the next thing is what is the goals of Human Genome Project. So if we see the goals of Human Genome Project, we have to find out that how many genes are there. So identify those genes, that what are the sequences over there which is present, whole sequence what is present, to store the information, to help the data analysis tool which can help in working us, transfer this technology to other sectors to make the helpful things for the humans, and address the ELSI, that is what? Ethical, legal, social issues with the project. Okay, next the method is ETS method or sequence. EST method means what? EST means that express sequence tags. Means what we that? We break the DNA molecule into parts with the help of restriction endonucleases and express that in the vector. Which vector do we use? We use either bacteria or the yeast. So bacteria artificial culture, yeast artificial culture, which help in doing it. Then if we are not doing this method, other is sequence annotation, means what? From one nucleotides to the end we are doing it. So this is another way. This way we come to know about certain features of it which you have to remember, which is given in NCERT. What are they? That human genome consists of 3164.7 million base pairs, average gene sizes to 3000 bases. Largest gene is dystrophin, having 2.4 million bases. Largest chromosome is chromosome number 1. Smallest chromosome is what? Is your Y chromosome, which has this number of genes. You have to remember. Next point is that total there are 30,000 genes. Earlier it was estimated 80,000 or 1,20,000 genes, which come to know that most of the part is free, empty, not nucleotiding anything. Okay, so less than 2% is actually coding for something. Then almost 99% of the nucleotides are same in everyone. 0.1% is different, which makes what? Polymorphism, which is a feature of DNA fingerprinting. Next thing is largest portion of the genome is repeated, which tells us about the chromosome structure, its dynamic properties and the evolution features. And around 1.4 million base pairs are present, which has only single base differences, and this is what? SNPs, that is single nucleotide polymorphism. Moving on to the next thing is DNA fingerprinting. In DNA fingerprinting, we will do what? We will identify the differences in the specific regions in the DNA sequence, and this is called as what? Repeated DNA. Okay, because it is keep on coming again and again in an individuals. So this DNA, basically the whole genome, if we are talking about, it is divided into two parts. What are the two parts? Number one is bulk part and a repeated part. The bulk part is the major part and the repeated part is the smaller portion of it, which we have further divided into what? Into the microsatellite, mini satellite and the etc nano satellites also. Now this micro mini satellites, that is different in different people. Me is different, you is different, that person is different, mama is different, papa is different. So these are called as what? This is called as polymorphism. Now we use this technique of finding the DNA fingerprinting is by the Alex Zafri. If you remember about biotech, it is pretty much same. 6 steps are given in NCRT, go through it. Over here they have talking about what? They are talking about the VNTRs, that is what? Variable number tandem repeats, which uses what? Southern blotting technique. Means we take the DNA fragments, isolating the DNA, cutting the DNA, hybridizing with the help of gel electrophoresis, finding the sequences. Then we are doing what? We are like denaturing it. And during denaturing it, we are doing what? We are taking the segments differently and put the radioactive isotopes, which will actually match with it and join with it, and we are able to find it out. This is the basic feature that we are working. Here they are talking about the microsatellite, that VNTRs are basically what? These are the mini satellites that are present, which size could be in the range of 0.1 kb to 20 kbs, which you have to remember. Now over here they have said it. So if you have to find it out which is the culprit, so this is your DNA that you present, you have to find out whether it matches with A or B. This is given in NCRT. Then you see that this fragment and this fragment, meaning this is not meeting. Okay, no fragment is here. Here the fragment is there. Another thing, this is meeting because it also has. So B seems to be more meeting compared to C. And now you will say that there is a certain portion where B, like C is meeting with the B also and A also. Of course, these are certain things which are similar in two people, like the skin color or maybe some other thing. Examples. Let's just give you one example on this basis, that is this. So this is the sample, we get it from the crime scene. Okay, so different locations are being given, okay, of the genes. So this is one location, another location of gene, another location of the gene. Now there are 3 suspects, A, B and C. You have to see this and find it out who would be the like possibly who could be the final guilty person of this. Okay, because this is the sample of criminal only. So you have to tell me in the comment section 1, 2 or 3 which one would be according to you is the culprit. Answer will be sent to you. I will give the heart the person who will give the correct answers. Well, that's all for this video. I hope you like this video. It's a longer video. I hope you would have able to understand or get the crux of the whole chapter in the one go. If you like this video, give it a like, share it with your friends and subscribe if you haven't yet. Thank you so much.